This is the I guess the last session of the conference, our shortened and online version. We have three presentations, starting off with Alastair Ross from AgResearch, and he's going to be presenting some interesting work done on the REIMS tool for rapid screening of sheep milk. So I think Alastair's with us. I think he is. Maybe he's not just at the moment. We seem to have lost him. No. So, and then after Alastair, we're going to hear a genetics update. So Suzanne Rowe from AgResearch, who is New Zealand's expert on genomics and genetics programs that she's involved with. So she's going to give us an update on that, and then Geoff King from MPI and I will sort of round off the afternoon in terms of presentations with the results of our survey of the sheep milk producers at about 12:40. So I think Alastair's with us, which is excellent. And so, Alastair, do you want to? Alastair is at Lincoln in the AgResearch team there, so he's going to look at. He's doing some work on sheep milk, or at least milk analysis. So are you all good to go there, Alastair?
Hello everyone, kia ora koutou. So I'm going to be speaking about a new instrument we've got, which has really been developed initially for human surgery, but then we've used it for meat, and now we've applied it to sheep milk, and it gives a very quick fingerprint, very detailed fingerprint of biological samples. So just first step really is why we want to get detailed information about something like milk, and we already know about protein, fat, and lactose, and these are quite easily measured. And what we can see is that we'll just get a point going here. What we've heard, I think, over the last few hours is just how important the minor components of milk can be. And we know that if we play around with things like the genetics, your farm system, your feed, environmental changes, that's all going to have an impact on things like flavour, processability, even micronutrient composition. And that's going to sometimes impact on protein, fat, and lactose, but more often than not, it's going to actually have a much bigger impact on these thousands of low concentration molecules, which can have subtle effects on things like flavour and processability, but also, quite interestingly, maybe also help to explain things around, say, origin, and also could be used to help pick up fraud. So we have a new tool at AgResearch in Lincoln. I'll just go this part here. If we're going to be looking at these detailed compositional aspects of milk, we have a lot of tools to do that. We have a lot of fancy mass spectrometers, and you'll have heard Evelyn talk about proteomics and mass spectrometers, and we have other ones which look specifically at the small molecules in milk, but it takes a lot of time and it's expensive. And this is a really big barrier to going into a lot of details. A lot of reason why I think we do stick to basic things like protein and lactose and fat. The thing is that maybe we don't need to be spending a huge amount of time and a vast expense on measuring these small components. Maybe we can do something that's a little faster and cheaper. And so what I want to talk about now are some results with an instrument called a rapid evaporative ionisation mass spectrometer. And handily, I'm going to show a video which our communication colleagues have put together. And this hopefully will work. Here we go. So it just demonstrates a simple measure. So this is basically just pipette some milk into a cup, use a surgical knife, and you basically heat the milk up, and that creates a vapour. It goes in through a tube, and boom, we have a mass spectrum of several thousand features, which we can then interpret. With a nice backing track there. So basically, just to summarise again, we just heat up a sample using a surgical knife. The vapour from that heating goes up through a tube and into a mass spectrometer, and we end up with a fingerprint like this. So this is just the mass spectrum of bovine milk, and we can see, you can see hundreds, but there actually are thousands of small peaks here, which are masses of molecules within the sample. So normally we'd be picking up a few thousand different, what we call metabolic features within a sample of milk, and that might represent sort of classes like fatty acids, or it might represent various lipids like diglycerides, triglycerides, and phospholipids. And lipids are quite interesting because they are very strongly influenced by things like diet. So in this case of sheep feed. And we've been using this a lot for meat, and I think this gives some nice examples of what we can do with REIMS, which could then later potentially be applied to milk. So, for example, we've used it to characterise what country of origin lamb has, and sort of, okay, we expect maybe you can find differences between New Zealand and British lamb, but we actually easily found differences between Welsh and English lamb. We have, within several different farms in the same geographic location, been able to detect the difference in kind of feed. So not just whether an animal's been fed on pasture versus grain, but actually what kind of pasture. So the difference between, say, plantain or clover or normal ryegrass pasture. We've also been able to use REIMS to follow in real time the evolution of flavour molecules coming off beef and lamb, and just sort of follow how these are changing over time. So again, sort of connecting then to the consumer, where we've been looking at consumer data on liking of lamb from different farming systems, and then connecting that with the molecular composition of the lamb as measured using the REIMS instrument. And we're able to find various molecules, which then we can then use to understand what is it about the lamb in this case that consumers seem to like or dislike, and how can we actually go and change that farming system potentially to go and improve that flavour. So, of course, we got a hold of some sheep milk samples thanks to Linda. We got chatting one day and said, it'd be great if we could maybe look at some sheep milk samples, and then we got approval from the sheep milk providers that have been involved in the MB sheep milk program. And so we took samples from a couple of farms, about 150 samples. It took a couple of days to analyse them, and then we just had the data to play around with. And obviously the first thing to do is to look and see, is there a big difference between ovine milk and bovine milk? And again, several presenters have spoken in some detail about the differences between ovine and bovine milk in particular. Under a mass spectrometer, they do look quite different if you look in detail, and just a quick explainer to about this kind of plot here. So here you can see that we've got nice separation. All these dots here, the green dots in this case are sheep milk, and the blue dots are the cow's milk. And each dot actually represents the information for one sample, and that's information are the thousands of molecular features that we've detected using the REIMS. So behind each dot are literally 1,000 to 2,000 different molecular features. So this is a very handy way of getting a big picture rather than just focusing on one or two different molecules, looking at the overall difference between samples based on their detailed molecular composition. And so in this case, we could see that we had around about 600 molecular features that differed between ovine and bovine milk. We haven't yet delved into in a huge amount of detail, but we have seen that, for example, a lot of these compounds are lipid compounds and will probably be related to both differences in feed, but also pasture digestion, etc. So there's certainly a lot to dig into there just as to why ovine and bovine milk differ beyond sort of fairly obvious explanations. So from these 150 sheep milk samples that we analysed, we were able to look at some data which compared different farms, so same geographic location, but different farms, different breeds, and we could see that there were 90 features that differed between Farm 1 and Farm 2 in this case, up around the Taupo Waikato region. And we could also compare breeds or sheep that have been selected based on their ability to produce high amount of milk versus those that sort of, I guess, produced an average amount of milk. And we could see that there were differences between these, even if there was not part of a specific breeding program at that point. And we can also see differences between early versus mid-lactation milk. So we can see, as is well known, that there are quite a few differences between lactation stage and the composition of milk. But of course, all these comparisons are not nice, clean, scientific comparisons. These are complicated by differences in breed. Lactation stages, we can see, has an impact, but also different kinds of feeds and different kinds of farming systems. So it's, we say, quite dirty data, but even with this dirty data, we are still able to see quite a few differences between the different kinds of sheep or farming systems, the sort of type of milk that's being produced. So there's a lot of information here that we can then use to try and see, okay, how can we understand why sheep milk might be performing differently when it's being processed, or why people might prefer one type of sheep milk over another. Another comparison we could do within this data set is we could look at the difference between sheep that have been housed part of the time in a barn, full-time in a barn, or full-time out in pasture. And when we looked at all this data, this plot basically shows that there is no difference, even if we're really trying to separate out the different kinds of milk from the different being inside, outside, or halfway through, especially those that sort of spent half the time in the barn and half the time on pasture, indistinguishable from the two other groups. However, if we do look at the same samples, but we remove those samples where they've spent half their time in a barn, we actually start seeing some quite large differences. In this case, 100 features differing between the milk that were fed inside and those that were outside. And of course, that will be related to both the fact that they were inside and the fact that they were being fed different pastures. So those kinds of differences are clearly coming through in the milk. So just a very quick summary. Using rapid evaporative ionisation mass spectrometry, we've been able to detect differences between different farms based on sheep milk composition. We don't really know what these mean in practice, and I think that'd be very interesting to follow up. We now have a fast method for getting this kind of detailed information. We can clearly pick up difference between bovine and ovine milk, which could be quite useful in trying to pick up fraud, for example. Someone's been trying to work off bovine milk powder as sheep milk powder, or as bovine feta cheese when it's as ovine feta cheese. So there's lots of potential applications there. There's a lot that could be done to understand differences in terms of seasonal variation, genetic variation, and geographic variation, to see how much control could be made over different kinds of aspects of the detailed milk composition. But one thing which is really, we think, is very interesting is in a future world where we don't know what we're facing in terms of environmental changes, by getting mass spectral fingerprints where you've got thousands of molecules that are being detected, you could actually use that to build databases and really understand changes based on changing environment, changing conditions. So you can go back and you can mine that data and try and understand, okay, in this particular circumstance, we've got an environmental change. We don't know what's going to happen to the milk. Let's go back and mine that data and see if we can predict what's going to happen to milk composition and that effect on milk quality. And then finally, I showed you in the video, we had a sort of handheld device where actually we're looking at improving that. We've just recently got a laser which makes improves the precision of the measurements and makes it much faster. So it's just an example here. You can just sort of see the laser dot there used on a plate of meat. But the same sort of thing can be done with milk, and it just improves the reproducibility and the speed, and it makes it a lot less hard work for the poor person who's actually doing the analysis. So with that, thank you very much for your patience, and thank you very much, especially though to the people who have provided the sheep milk samples and taken that time, and they've been very encouraging in terms of their interest in doing more science around sheep milk. Thank you.
Fabulous. Thank you so much for that, Alastair. That's really interesting. And I can see all kinds of, I mean, it'd be great to sit down with a whiteboard and just plot out a whole bunch of really interesting options as the things that you might want to take a look at and where that might go. So yeah, that's a fascinating, I guess just a question, if any, I'll just throw in at you now just before we flick over to the next presenter, but based on your experience of working with meats and the different profile of that meat during a season, for example, or during the age of a lamb, what can we expect in terms of being able to tell, you know, the appropriate sort of, or the best possible quality of milk for particular kind of diets or for particular kinds of products, or even, you know, going into particular kinds of, I guess, health responses, which I was just thinking earlier about how this relates to both Amber's work, but also to Diana's work on environment. So I think there's a whole bunch of issues. And sorry, that's probably not one question, but I just think you've got something interesting going on there.
Well, I think that's you've struck on probably the best thing about this kind of approach is, one, it's very simple, but two, really the sky's the limit in terms it's completely up to you in terms of how you want to look at that data. And so while now we don't have a huge amount of data, we've just looked at 150 milk samples, and we've taken the available extra data to go with that. But if, as part of a sort of a regular program of testing, you could then go back and you could check for anything. It really is, it's up to the individual, and I guess to come up with, I guess the sky's the limit in terms of the questions that they're asking. So you can sit there and go, well, I wonder what the effects are of seasonal duration, or of this particular weather pattern, or maybe if there's been sort of, it's a bad spore count year or something like that. Has there been any impact on the composition of the sheep milk?
Yeah.
So
precisely.
There's really, that's, as a, I guess, probably more an analytical scientist, I'm not the expert on how to interpret the data. It's really fun sitting down with the people who are actually involved with the samples and who know really about, in this case, say, the sheep milk industry. How, ask the questions, then we'll see what we can get from the data and try and make something from it.
Yeah. Well, I think both Spring Sheep and Maui might be interested in this, given they've both got very, very strict quality and says compositional kind of like parameters to work towards with their various products. So, I think this potentially has some really interesting flow-on effects for them.
Yeah, so I've got some comments here from YouTube. It's Billy Zheng, who says that very useful technique for research and industry. And Helena Pereira, who says really interesting research. So thank you for that, Alastair.
Oh, no problem. Happy to talk to anyone who's interested in finding out more. We are sitting on a lot of other results, which I decided to remove at the last minute for not wanting to rabbit on too long.
All right. So people can contact you via the AgResearch website, I guess, or they can. If you've got your presentation, I'll pop it up on the stream site in a minute. You get your email address via that, I guess.
Yep. I'm happy to have a chat.
Very good. Thank you very much. So I'll turn over to your colleague, Suzanne Rowe, who is sitting there in her lovely lounge in Twizel. The amazing thing about this, doing it in this environment, and you'll see that I've got a little background here that shows all the things that are not going on in my messy office at home. But it's very interesting beaming into people's homes, at this time. Not that I can't see any dirty dishes on your kitchen bench there, Suzanne, but you're in lovely Twizel. What's it like down there in Twizel this afternoon?
Oh, it's pretty sunny. It's generally pretty sunny here, actually. It's not a bad place to hole up for a few weeks.
No, well, I think you've got a brilliant spot, and obviously there's not a whole lot of people going past the gateway, I guess, on this time of the day.
No, I've shut the dog away. I've shut the kids away, so fingers crossed when we make it through.
Okay, so over to you, and you're going to give us an update on genetics and genomics and the work that you guys have been doing.
Okay, so, Thank you. Thank you for keeping going. Everyone else seems to have cancelled their conferences, and the dairy sheep industry seems more resilient than that, because here we are with 150 people listening to talk. So thanks, Craig and Linda, for all your hard work. My name is Suzanne Rowe. I work in the genetics and genomics department of AgResearch, and I'm just going to give just a quick update on where we are with dairy sheep breeding and the sort of the tools that we're developing, not just for dairy sheep, but other breeding schemes. So I'm going to talk a little bit about dairy breeding schemes, and the main focus for us is firstly what to measure when we're breeding for something. How to obtain a breeding value. So that's a value that's attributed to a parent and tells us how that parent's offspring is going to perform. And that's how we choose our parents for the next generation. That's the crux of a breeding scheme. Genetic gain, so how to go faster, how to make as much progress as we possibly can using breeding values or parent evaluation. What advanced tools are out there? What sort of genomic tools we can use to go faster or to do things differently? And finally, broader research in the sheep area around genomics, and that might be genomics of the microbiome in the sheep gut. It might be methylation of the DNA pattern within a sheep, or it might be how that sheep reacts to its individual environment. So whistle stop really. But the first thing that I'm going to address is variation and why we want a breeding scheme in the first place, because a lot of people are sort of looking at breeds all over the world and saying, well, hang on a minute, the easiest way to improve dairy sheep production in New Zealand is to import a high producing breed. And of course that's a big step, and it's the first step for a lot of breeding programs. But I just want to show here, we've got some French data here where the variation between breed and we've got a lot of ewes breeding, and we see an average of 162 litres versus 317 litres in the Lacone. So it's pretty obvious that if you can feed and maintain a Lacone, that might be the breed that you go for. But I just want to look over here at the standard deviation. There's 100 litres standard deviation within these Lacones. So what that's telling us is that in actual fact, we might have a Lacone giving us 200 litres, or we might have a Lacone giving us 400 litres. So it's still really important to have a breeding scheme within the flock or within the breed, as well as choosing breeds that give us more performance. And that's really what I'm going to focus on today. You've chosen your breed. How do we make sure that within our flocks or within our breeds, we choose the right parents going forward? So finding the best animals, of course, not just what we see. There are feeding changes, management, environmental perturbations. There are differences in health between individuals. So the first thing that we do is we compare animals within a group. So we need to compare animals that are fed the same and managed the same. And that sounds obvious, but it really is where most breeding schemes fall over, is just inaccurate comparisons. And we use relatives for information. So when we look at an animal and we look at its genetic performance or how we expect it to perform, we don't just look at that animal, we look at all its relatives. So the more information we can have on family, on relatives of that animal, the more accurately we can say how well it's going to perform as an individual and how well its offspring are going to perform. And really it's down again to this breeding value. And that's the part of the performance of the animal that will be passed on to the next generation. That's really shown here. So this is a plot of phenotypic deviation. So that's basically the litres of milk above average that these individual sheep have all been given, so above or below. And then there's the breeding value. So that's what our expectation is, that these sheep will pass on to the next generation. Now, these two animals here, this one and this one, the two that are ringed, basically have the same performance. So they've given the same amount of milk in this generation, in this lactation. So if you were choosing them based on your lactation records, you'd pick them both and you wouldn't know how to choose between them. But if we look at their breeding value, so that takes into account everything else that's happened to them over the year, that takes into account their age, how they've been fed, and it takes into account family members. In actual fact, one of them would give you offspring that gave you about a litre less than the average in your flock at the moment, and the other would give you almost 15 litres greater than what you're seeing in the average of your flock. So if you don't have that sort of background breeding information, then you really can't make the choices that you need to make the progress that you would like to see. So it's all about really successfully choosing the best parents and the top of the sort of the distribution in your flock, choosing these ones at the top here so that you can make progress. So AgResearch have been building a pipeline for a few years now. We've been working on, firstly, summarising milk data, trying to work out how we record data, what's the sort of most useful data point that we can have. We've been looking at what we need to adjust records for, what's important to a dairy sheep scheme. So lambing date's important, the number of lambs born is important, the group that they're fed in, of course, is important, live weight and health traits. And we've sort of catalogued the ones that are most important, that really need to be recorded for a successful breeding scheme. We're using weekly averages. We found that was actually the most useful and the most informative piece of information that we could use to build up a lactation profile. We're fitting a model. We've worked with various models and worked out one that we like to fit that basically fills in the gaps. So if we've got weekly profiles or even monthly profiles, we can come up with a lactation curve, and then we can come up with an estimated total yield for an individual sheep. We fit genomic relationships or pedigrees, so that means we look at all the relatives as well as the individual, and we obtain breeding values. And that's what we've been working on over the last few years, and we've done a lot of work with spring sheep. And now we're moving on to the next part, which is what do we do with this? So as a genomics researcher, I can estimate breeding values for farms in New Zealand, but of course that's not very effective or efficient. And there's a database out there. Sheep Improvement Limited or their new setup, Improve, which basically does that for many, many traits and has done for a long time very successfully. So they're already producing breeding values in the sheep industry for many other traits. And they're doing a great job, and these breeding values have been used for all sorts of research. So they're the absolutely obvious platform for us now to pass on the work that we've been doing and the knowledge that we've gained, and for them to basically take this out into industry and offer everybody the opportunity. And many sheep producers will be using SIL or Improve anyway, so to add on milking data is an obvious. So at this point, they're not producing milk breeding values. What they have done is they're set up to take the information, they're set up to take the data. You can register with them, you can pay for your data to go in. And can you still hear me?
Yep.
Yeah.
All good.
Okay, good. Okay, my computer's telling me my connection is unstable, but we sound pretty good so far. Okay, so basically you can put your information into Improve, and it can be manipulated, it can be adjusted, it can be reported, you can manage your data, and from then on, breeding values are going to be produced. So I've put a name here, Sharon McIntyre at Beef and Lamb. She's the person to talk to about this. She's the person that can help with anything that, if you're interested. So if you want to get hold of me, please do, but Sharon is the person that will be able to help in the future. So part of Improve, or part of the big difference between the old Sheep and Lamb Sheep Improvement Limited database, so SIL, and Improve, is that they've added genomic information in a different way. So they've managed to absorb genomic information into their breeding values. And this is just a graphic for a particular situation. Accuracy is basically how well you can identify the parents, and accuracy is linearly related to how quickly you can progress. So if you are identifying your parents literally by eyeballing them, you're not going to make much progress. If you've measured one parent really well, you're going to make some progress. If you've measured two parents and you've got breeding values for them, you're going to make even faster gain, and so on. These green arrows at the top are what happens when we add genomic information. So when we add genomic information, we even more accurately identify parents than if we're just measuring and recording. And what that does is help us to go faster. So that's really the crux of what we've been working on, how we can add genomic information to a dairy sheep evaluation scheme. Anyone can do this. Anyone can get genomic information for their flock and incorporate it. Getting genotypes isn't a difficult thing, and it's not actually an expensive thing either. So this is the process we have in genomes at AgResearch, but there are other laboratories available that can genotype for you. And it's literally when you take an ear tag, there's a little tissue sampler here, so a little receptacle that takes the little piece of tissue that's displaced when you pop your ear tag in, and it's barcoded. And here you can just see a sheep being ear tagged, and it comes through and it goes through the lab with its individual barcode through a bunch of robots that extract the DNA from that ear punch, run it across some chips. So these are platforms basically where a whole heap of DNA variants have been stuck to them, and what they do is assay or test your DNA as it gets flushed across it. Generally, we're looking at around 15 to 50,000 variants or DNA changes, and that basically can be used to produce genomic information. One of the most important things for us in New Zealand is that most of these chips have been produced by an international genomics or sheep genomics consortium, and most of them were developed in New Zealand and still are being developed in New Zealand. So we actually have the power to design these chips in exactly the way that we want them to give us the most information and to be the most useful. That's one benefit. The other benefit, of course, is that everybody else across the world who's got research programs that are identifying variants that are really important for sheep research actually are then placing them on here. So we get the luxury of getting the results of everybody else's research placed on these chips too. So we're in a pretty fortunate position. And at the moment, for 18,000 markers, it's around about $25 to get genomic information. So why bother? Genomic information gives us relationships, it gives us relatives, and of course we were getting that anyway from pedigree. But pedigrees often don't give us everything. They're basically relying on us writing something down, recording and observing something. This is 791 odd records that were just pulled where animals had pedigree and genomic information. And there's a lot of zeros here in the pedigree information, and that's where there was wrongly recorded information, so animals didn't look related. And then when you do a genomic test, actually they are related. So that kind of whose baby comes out really well with the genomics. So first of all, adding genomics gives us pedigree relationships that we didn't realize were there because they were wrongly recorded. Then genomics gives us precision. It gives us absolute relationships. Generally, in a pedigree, we might have a half SIB relationship, which is a quarter, or a full SIB relationship, which is a half. But in actual fact, it may not be a quarter. It might be 0.31, or it might be 0.4 to 0.6. So it gives us a bit more precision in our relationships, which again gives us the accuracy to go faster. And finally, if we have a single point mutation in a particular gene that we're interested in, like Scrapie or BLAD or CVM, then genomics gives us the opportunity to actually pinpoint individual animals that are carrying different variants. So as I said before, we are part of an international sheep genomics consortium, and these dots are individual breeds. And when we have genomic information, what we see very clearly is that individuals within a breed group very closely together. So if you have an animal of unknown breed, you can simply put the information into a map like this, and it will pop up in whatever breed cluster that it belongs to, or maybe halfway between two if it's a half-bred. So it's a really nice way of being able to tell what sort of breed proportion you have in your flock and where animals sit, as well as how they relate to each other. So another thing that we've been doing with genomic information is that we can actually control inbreeding much more precisely. So these are maps of sires and basically looking at how they relate to each other. What we've done here is chosen the 27 highest index sires, so the highest breeding values, and we've got an average of 6 in the index. But actually, these sires are really quite related to each other. So if we just did that, if we only chose the top sires, what we end up with is increasing the inbreeding in our flock, and that can lead to genetic determinants that cause problems. What we can do, though, is we can balance. We can use genomic software to put the genomic information in, and we can say, give me the best index that I can get whilst keeping my inbreeding coefficient below a certain level, so we can cap it. We can say, I only want this much inbreeding in my flock, so what are the best sires to choose to give me the maximum progress I can make? And we've been working on this for not only sheep and dairy sheep, but other species too. And it's a really effective way of maximizing your program, but keeping your inbreeding to a sort of a sensible level. Major genes, as I've said, genomics also gives us the opportunity for point mutations. So this is a French study that was done recently. There are many, many more studies out there where people have already identified variants that are exciting, they're interesting, and they give us more information within our breeding schemes. And we can add that onto the genomic chips, and we can use that information alongside everything else. So we're also involved in a few international projects. So we work closely with the International Committee for Animal Recording, and that's really important. So as when we measure things, or when we decide how to measure things, that they are comparable globally. I think we're going to become more aware of what's going on around the world and how we compare. So it's really important that there's a standard, there's an international standard for how we measure traits. We are part of something called Grass to Gas. That's a new global research alliance program, which involves 8 partners across the world. And basically that's looking at feed efficiency in sheep, that's looking at environmental impact and resilience. And as New Zealand, AgResearch has joined this as the New Zealand partner. And we're also affiliated with the Smarter project, which is another European project, which also looks at efficiency and resilience, and that has a strong dairy sheep component. So within AgResearch, we're working within these programs, and anything that basically comes out that's applicable to dairy sheep, we're trying to bring it back, and we're trying to incorporate it in the advances that we make and that we roll out into industry. And finally, just some other research programs that are applicable. We have an MB program, or an MB funded program, sorry, working in the methylation space. Methylation is I talked to you about animals having variants that change their production. Well, actually, those variants can be switched on and off by environmentally induced switches, and we call that methylation. And we've been looking not just at the actual readout of DNA on those chips, but actually the expression or the switches that switch those readouts on and off, and how they vary between individuals, and how that might be affected by stress or environmental changes. So that's some really kind of blue sky fundamental research, but actually that can have a really practical outcome in the dairy space. We've been looking at microbiomes. So we've talked a bit about microbiomes in humans and how they digest sheep milk, but actually we're interested in microbiomes in the ruminant itself. Ruminant microbiomes are really interesting, and they tell us a lot about the animal. They tell us about the environmental impact of the animal. They can tell us about how that animal ingests and copes with feed. And health and all sorts of things. So we have programs looking at microbiomes, and we've been taking microbiomes from dairy sheep and seeing how they differ to meat sheep, or how they might differ. We've been looking at environmental impact. We've been looking at methane. We've been looking at methane in dairy sheep flocks. And we've also been looking at methane in meat sheep flocks, and we've been looking at the milk of animals that are high and low methane, and using tools like the one that Alastair talked about to look at milk differences and how that might be related to how the animal digests its food and how it produces greenhouse gases, and how that might affect its environment. We've been looking at feed intake and efficiency, and we've been looking at digital monitoring systems. So how we might use things like GPS collars to assess how much an animal's eating and choose animals that naturally are more efficient. There's individual variation in how much feed an animal needs for maintenance or milk production. So there's a lot of research programs going on that are coming through. But back to what we're doing at the moment, we are working on basically moving the dairy sheep sort of breeding values that we've been using into a national module that anyone can access in a standardized and structured way, which means basically that you can register, you can get regularly updated breeding values for your flock. These analyses are often run weekly or fortnightly. So they're in real time, which is really important in breeding season. You should be able to compare your flocks with others. You should be able to adjust for genomic breed, and on top of that, we can offer enhanced tools for genomic analyses. And as we work towards getting more and more data, one of the things that the Sheep Improvement Limited database has done really successfully is get data all in one receptacle that then can be used to look for things like major genes. So researchers have access to a large body of data that can be used for national good. As I've said, there are new enhanced chips at AgResearch. It's $26 an animal for 17,000 markers at the moment, but there are other sources. And I'm quite sure that it won't be long before we are looking at cross-country evaluations. So that's just a snapshot of where we are at the moment. This is the bunch of people that I work with when I'm not at home. As I say, we've had a lot of funding from MB, and thanks to Spring Sheep, to Southern Cross, and to Barry and Judy Foote, who've been helping with goat research, which is very closely aligned to the sheep research. And that's me.
Fabulous. Thank you so much for that, Susan. That's really really great coverage of much of this really interesting work that you guys have been doing. There's some questions which I'll just sort of put to you, and so one of them is, how does one of the questions from Richard Shorey says, how do the health traits, especially FEC, which I guess is facial eczema, relate to milk traits? Have you got any commentary on that at all?
Not specifically, no. I mean, there's been a few talks over the years at this conference where people have shown that they've seen a milk drop, if they have underlying facial eczema. And it's certainly something that we're interested in with the microbiome work and with the methylation work, because it's a really important subclinical stressor. So we're constantly looking for other ways to pick up that subclinical effect. But I don't know the direct link, or if there is a direct link between facial eczema and milk constituents.
Right. So just a couple more questions. So just in terms of cross-country comparison with China, is that something which is on your agenda, or is that part of the work that you were talking about, international collaboration?
So we have been involved in helping some Chinese researchers to set up progeny tests. We haven't discussed comparisons. I think the first comparisons will probably be with either France or the UK, where a lot of the imported animals have come from, and we actually share connectedness and linkage. So it will depend on where the animals are that we share most in common with, and where the systems are that we share most in common with. But there's no reason why we can't have a cross-country evaluations with any country, providing that the people are willing to put their data in the same place and compare it with the same background. Right.
Actually, my mistake. It wasn't facial eczema. It was fecal egg count. So that's sort of the loading in comparison with milk traits. So I'm not sure how that would relate to the work that you're doing, but if you've got a comment on it.
I don't know how worm burdens affect, other than obviously depression. Generally, if an animal has a high worm burden, then it's unhealthy and everything suffers. what you can do if you use something like Improve is that if you have a breeding value for worm burden, you can actually incorporate it into an index with your milk traits. So you can look at them, simultaneously. But I don't know what the direct effect is.
Yeah. So just a question from Helena, who I've had a little bit to do with. So I think she's on a trip around New Zealand and talked to some farmers. But what would you see as the key question is, what would you see as some of the barriers for farmers, maybe not in the sheep dairy industry, but more broadly in the dairy industry, in using some of these tools? Because there is a lot you're offering there, isn't it?
I think probably the first barrier is actually recording the data, and getting it somewhere where it can be handled and used. And these national schemes sort of help with that, because generally that's mediated through a bureau. So you give your data to somebody who enters it on your behalf. So that's one of the big barriers, is being able to measure, and be that by milk meter or herd test, and measuring mobs, and just getting the measurement right. In terms of barriers to genomics, I think a lot of it is just not really engaging. So it's there are many industries now where having your applicator and your ear tags and your TSUs is pretty normal. So that's just us getting out there and helping people with tools and price. I mean, that the more that we can drive the prices down, so as they're accessible, so they become sensible, then the better that people can adopt and uptake.
Well, I mean, I don't think price is particularly an issue, given we're trying to all push up production in the dairy sheep industry. And if the prize is $1,000 animal, then I think $25 per for a chip is not a huge sacrifice to make. Final question, I guess, from me is I guess what sort of take up have you had across the sheep dairy industry? I know you've worked closely with Spring Sheep on this, but what about the smaller producers? We've got sort of 15 or 16 small producers. Are they interested in doing this? Have you had some contact with them?
We've had a bit of contact, and we're working with a couple of people. I think people are gradually moving from the idea of pedigree beefs to BVs to genomic BVs. I think people, as they set up, are starting to inquire, and basically we're at the stage now where a lot of small producers are getting their houses in order. They're getting organised, and once they've actually got a shed built and they're milking, then they'll be looking to make the next step. One thing that we can do is when we work with the bigger producers and the people that are probably going to be supplying REIMS, of course, we benefit everybody right down through the chain, and that's something that's really important. And go on.
No, I was just going to say, that's great. So just notice on the last slide and some of the sort of the future-facing work that you're looking at, it's very much environmentally orientated, microbiome, methylation, that sort of work, which suggests that actually part of the challenge here is not just the animal measurement, but actually looking at really closely at interaction, and particularly being able to reshape the animal as a response to their engagement with particular sort of conditions and things. That sounds amazing, I guess, and challenging for genetics and breeding more generally.
Yeah, I think resilience is going to be really, really important, particularly as we see these big environmental swings, and a lot of people are really concerned about that. We've shown that we can breed for production, and we need to keep our eye on production, and we need to keep pushing for it, but we really need sheep that stand up to the environmental conditions that they're in, and it's really important that we have tools that can monitor that and pick up those sheep that might be more susceptible to environmental stresses.
Yep, cool.
So
fantastic. Nice stuff.
We have a final question here. It's a follow-up on the earlier question about if we can work together, say New Zealand and China, to do genetic improvement. So the follow-up is saying dairy sheep will most likely be housed in China, but most will be under grazing conditions in New Zealand. so how does that affect kind of the data that we get from the SNP chip with these different conditions in different countries?
Yeah, really, it's about G by E and whether animals rank the same. So when we look at genomic breeding values, we're not looking for absolute differences in the animals. We're looking for whether they rank. So we want to be able to choose the top animals. And often it doesn't matter which system you're in, the top animals rank the same. So where the research is key is just looking at the house animals, looking at the grazing animals and saying, do they rank the same? And if they rank the same, then we can use the same analysis. And if they don't, then we have to fit some sort of component that accounts for the fact that they don't rank the same. And of course, that's crucial and it's key and it's part of what we do. So I'm not necessarily advocating that we rush out and use data from China. I think we use data that firstly is most applicable to what we're doing and most comparable to what we're doing, but that doesn't mean that we can't use other data and that it won't still give us information, even if it's not using exactly the same system as we are. We just have to be aware of the limitations. Okay, thank you.
Cool. Brilliant. Thanks very much, Susan. That's fantastic. Good stuff. Yeah, really appreciate that. So it's a really nice contribution to the overall sort of day. So nicely done.
Thank you.
Okay, we're moving on to the last formal presentation, at least. This is, it's me. No, it's not. It's Geoff King. So Geoff is from MPI. He is Senior Advisor with the Market, I think I get this wrong every time, Geoff. Is it Market Intelligence? But it's something like that.
We work in the Market Insights team, which forms part of MPI's Economic Intelligence Unit here at MPI. Let me just try and. Now, you showed me how to do this yesterday, Craig. Bear with me. Have we got the? Is my screen showing?
Good to go.
Okay, thanks, Craig. So, just to finish up today, what I wanted to do is present some of the preliminary results of the first ever survey of sheep dairy farms in New Zealand, and this was a joint project between MPI and Massey University, and it was led by Craig, yourself, Craig Prichard, and my colleague Anne Nguyen in the Market Insights team, who I think is on the call, but unfortunately couldn't present today. Now, just a little bit of context on who we are. Let me just. So Anne and I both work for MPI's Economic Intelligence Unit. And one of the roles that our team has is to look into new and developing industries. We also look at consumer trends that are likely to present sometimes challenges, but also big opportunities that we see for New Zealand's primary sector. And the emerging sheep dairy industry certainly presents an opportunity that we were keen to investigate. And at a broader level, the EIU also funds within industry and collects a lot of data around farm level production, financial information across a whole range of industries. And you can find reports and that data on our website, along with our quarterly publication, the Situation Outlook for Primary Industries. So just sort of to give a bit of background, the genesis of this survey occurred when we attended last year's conference. We chatted to you, Craig, and a few stakeholders. And it was prompted by the fact that there's quite a genuine interest amongst our policy colleagues here at MPI in the potential for this sector. And we sort of felt that if we could meet with industry participants, sort of gain some insights into their experiences and publish that information, we hopefully could raise awareness of the sector's growth and potential, and we sort of hope that the data and the final report, when it's produced, is going to be helpful in that context, not just for existing operators, but potentially for new entrants, and also to our colleagues within MPI to support, their future policy development and ministerial advice. And before I sort of start showing you some of the the findings, I just want to say a huge thanks to all those farmers that gave so generously of their time to help us with this project. Really, really appreciate that contribution. We ended up interviewing all 18 commercial farmers around the country, and asked a huge range of production and financial and systems-based information over a period of about 3 to 4 months. And so perhaps maybe the starting point is a bit of context of where we're at now in terms of the industry. And Craig, I don't know if you want to sort of talk to the current state of the nation in terms of the industry.
Yeah, will do. Thanks, Geoff. So just to, I guess, fill in, and this is not news to anyone that's involved or has any sort of general sort of feel for the industry. We have a large cluster of both animals and farms in the Waikato, and we have also, a group in the sort of what we might call the East Coast Wairarapa sector of New Zealand. And then the third sort of cluster is in Canterbury, which is where obviously we had hoped to have the conference this year. There's quite distinctive differences in the profile of those different clusters in terms of the size of the operations and their the sort of the development, the products they're producing, and obviously there's some quite interesting differences between the sort of animals and breeds that are there. So, but just to be clear, we went around all 18 producers. With the results that Geoff we're going to run over in this presentation, aggregate those, and we've just tried to pull out some of the key points that overall we're interested in, and I think everyone would be interested in terms of going forward. So I think that's just a general overview. You can see some of the numbers there. You go.
Yeah, I think it's an industry that's small, but certainly growing, relatively rapidly. So let's look at a few of the key findings. Now, we're sort of working through the data now, and given that we've only got a few minutes, we're sort of keen to show you maybe the edited highlights, some of the key themes or findings that we're seeing early in terms of going through it. Firstly, in terms of the motivations for entering the industry, I think unsurprisingly for the farming sector, passion for sheep dairy ranked highest. And also, the ability to gain price premiums, profit performance over time was also a consistent theme in that response. And, if we look at that third category, given the environmental regulatory pressures that are affecting the pastoral sector more generally, and especially, bovine dairy, it's probably no surprise to see that environmental performance is also a high motivation as well. We also asked our participants a range of questions around establishment costs, and I thought the results were quite interesting. There's quite a large variation. They're showing, depending on whether farms were either greenfield establishment or a conversion based on some existing infrastructure. And what was encouraging, I suppose, was many farmers were able to convert and adapt existing infrastructure at a relatively economic rate. I don't know if you have any sort of context there, Craig, or anything you can share on that side.
Yeah. So as everyone will know, that the sort of preferred option for everyone, really, depending on the sort of scale, is a conversion of an existing cow dairy operation. Now, that's not to say that a greenfield build is not, has some real strengths to it, but certainly keeping costs down and being able to provide, address some of the shift that people have converted existing cow dairy operations for, as you said, relatively economically. Some of those very economically, and I just want to make two sort of just general comments about that. We've a couple of the producers have actually made a very, very easy switch from an existing cow operation to a sheep operation, and I'm particularly thinking here of Richard Thorpe at Waimata Cheese, who has an extraordinarily rich piece of land on the flatland not far from Gisborne, and he got out of milking cows and put in a sheep operation and did it all himself, and did it in a really fantastic way. So it was a very cheap or relative in terms of actual cost, it was a really cheap switch. But it's a fantastic example of how you take an existing cow operation, you make an economical change to essentially putting in bales and getting and clusters, and you're away. And he really had and just significantly increased the returns from that establishment. When you're in very expensive flat land, I mean, Richard's right next to an orchard, which, obviously you want to make the best of every single hectare. It's a very, very cheap operation. But I would also, and we had hoped to, if we had had the conference in Canterbury this year, to take everyone on a trip to both to Matt Jones's place at Kerwee, and also to James Thompson's place just at Harewood, there, not far from the airport. I just want to talk briefly about James's conversion, which for me is an outstanding piece of Kiwi ingenuity, really. What he has is essentially a greenfield site. He's built a sheep milk parlour where it was using 4 containers, 4 sort of shipping containers, a plastic roof, which is obviously solid and stable and very really great for keeping the weather out, to put a really good piece of concrete down, and cobbled together a milking platform and various bits of technology using Reed's engineering from just up the road at Ringiora. And that is a fantastic conversion. Essentially just put it out there in the paddock. And it's really, if anyone was interested in what a greenfield operation would look like for a relatively cheap price, where he essentially bought and borrowed from various locations, then James has got a fantastic example to show. Matt as well, in a different type of way, but both of them have got some really important sort of just done it really well in a sort of very, very tight budget. And just finally, I would just note about those 2 producers. Both of them have built into their plant the ability to scale up very quickly. James, in fact, has got a 9,000 litre tank. There's no way he's getting anywhere near close to filling that at the moment with 400 sheep, but he's got significant ability to growth in numbers and scale. And that would be, we're hoping in the next little while that we'll get a manufacturing option in place for Canterbury, and that both these operators and others in the Canterbury area will be able to scale up and really get some returns rolling in from this new and hopefully more environmentally higher performance environmentally than the existing cow operations in the area. So yeah, that's kind of just my comment on conversion costs.
Craig, and in fact that segues quite nicely into our next slide, because we were quite curious to see what sort of scale the industry was operating at, currently, and these 2 graphs show a spread that reflects almost 2 different industry structures that are existing at the moment, and for those regions with access to powder processing, there is currently the ability to scale up in terms of size, while at the other end, there are those artisan sort of cheese and yogurt producers which are operating business models which are more suitable for smaller scale production. And so, if you look at that in terms of a supply chain structure, which we put together a little sort of diagram of how we sort of saw the industry in terms of what came out of the survey. It does feel like there's a sort of dual structure that the industry is currently sitting within, and that's sort of seen here in this simplified slide. We've got some suppliers supplying spring sheep and Danone, and with dryer facilities, the focus there is predominantly on infant formula and milk powder, and these are going to export markets, mostly in Asia and also into Australia. For the majority of farms who either make their own product or sell for contract production, the product mix is a little bit different. It's predominantly cheeses and yogurts, fresh milks, some flavored milks, and those products are currently being focused on the domestic market through a variety of channels. So two different sort of structures where the industry sits, and that's also regionally based, as as you say, Craig, and hopefully over time with more dryer facilities coming on stream, especially in the South Island, for example, we'll see that ability to scale and see that flexibility of product and market expand as the industry grows. One of the big themes that's emerged from the survey is sort of represented in these two graphs, or at least we've tried to show it, and it follows on nicely from Suzanne's presentation just before. It seems to us that the big focus for farmers right now is increasing productivity levels in terms of milk production. It's fair to say that at a national scale, we sit behind, perhaps our European competitors, and that's definitely reflects where we're at in terms of our genetics within the flock. Now, we weren't unfortunately able to come up with a genetic profile of the national flock, but the graph on the left gives at least a tabulation of the breed types that were cited by our survey respondents. So it's not the most scientific way of representing it, but it gives a flavour of where we're sitting now. And Craig, we were chatting about these results, and you mentioned that you're expecting that profile to change over time with more Lacoon coming through in the future.
I think everyone's been quite impressed with the potential of the Lacoon as we go forward, and certainly many of the small producers, particularly, have looked or bought some either REIMS or Lacoon genetics. So there's an AI work going on in that area, and that's not to say that the East Frisian genetic material or that breed is not important. And as Geoff said, this is a respondent-based overview rather than a flock-based fruit. So it shows, it really is kind of historical in it's looking backwards to where the New Zealand sheep dairy flock has come from. I just want to reiterate Geoff's point about, if we look on the right-hand side, so this is probably of most importance to people in the industry, and obviously we've talked about this over many years. The aim is to push the the purple to the right. So the deep purple slide is showing the current milk production per ewe over the 18 farms that we looked at, and then the lighter purple is the expectation in the future, and that is kind of where we're headed. So the expectation is in 5 years' time, with genetic game improvements in feed quality, and a actually much closer focus on, I guess, particular system development, then the aim is to get towards that 350 to 400 litres per animal. And we can kind of get a feel for where we're still at. We're still, the majority of animals are still in that sort of 200 to 250 range, which, is where we need to be in the 300 to 400 range to be really making some sort of some gains, both from economic returns, but also just being able to support the kind of investment that some of these businesses are carrying as they go forward. So it's encouraging, I guess. If we were to look back a couple of years, we may have had an even steeper graph, a deep purple graph line going in that direction. But I think we're in the midst. And just one thing, and Geoff will probably talk about this in a minute, is that the aim of the survey is that we'll do this perhaps in a more sort of efficient way over the next sort of few years, in order to really plot where that graph is going, because I think that gives us some confidence, and also gives us some ability to kind of promote to new entrants just what kind of returns that they might get in the next few years.
Yeah, I think that's an important point. I mean, as that productivity increases, it's not just going to help our new entrants. It's going to give confidence our existing entrants, but it's going to give confidence for new entrants to come in, and there's going to be strength in numbers as the industry builds, and is able to sort of build a more secure and growing supply. So the last thing I wanted to just show you was we asked a bunch of questions around challenges and advice for new entrants, and we see those themes coming through again that have come through in the early findings from our data. In terms of the challenges the business is currently confronting, the themes are, we need to educate our consumers about, how great our product is and its unique features. We need to focus on developing new products that meet the consumers' needs, and we need to focus on marketing those products. And in terms of building supply, the challenges in focusing on lifting those low milk yields that we're currently seeing, and also in key regions, trying to build processing capacity. And then when there was one of the biggest themes coming through in terms of advice to new people entering the industry is that bubble in the top left-hand corner. Invest in quality genetics. Get the right sheep. And also, I probably would draw attention to a lot of the respondents saying, get your product right and get your market first. Craig, I don't know if you want to sort of add anything more on that.
I was just reflecting a little bit on having talked to everyone about this, and when you ask that final question, what sort of advice would you give to people entering the industry, there's a sort of a pause, and everyone reflects a little bit on some of the pains that they've gone through in getting their operation up and running, the sort of things which they've struggling with and continue to struggle with. And I think it's the one that kind of comes through, it'll take longer and be harder than you initially imagine. I mean, I think we're in an interesting phase in terms of the industry. The first movers and the first, the pioneers have kind of made huge gains and really gone through some pains in order to get those gains, and they've laid out some interesting paths and some important paths for the new entrants. And I would just note, we're in the midst of quite a big shift at the moment. It's likely to be 6 new producers in the Waikato over the next 18 months or so. Both Spring Sheep and Maui are working very closely with some people that are very close. And just in the last sort of couple of months, I've had contact with new producers in the Canterbury region who will also be joining the group. And we are in an interesting and important sort of shifting phase. Again, access to processing facilities in the North Island is making a massive difference, and just building supply to match the demand which both Spring Sheep and Maui have identified in different routes is creating a lot of confidence and a lot of important shifts. But those pioneers and those people certainly outside of the Waikato, it's going to take longer and harder than you initially imagine, and product to market first is really the crucial message. Having said that, if we can just find, and this is perhaps the kind of one of the reasons why I really got involved in this project, is that anything which puts a little bit of pressure on the big institutional actors, whether that's the state or the banks or the big agribusiness operations around New Zealand, particularly the major dairy companies, if we can get them to take more interest and more notice in this, then we might be making a little bit more progress. So that processor in the South Island is the big holding things, the thing that's holding things back at the moment, as we know. And these comments reflect that. I guess the two, what would you call it? The two phases and stages that we're at the moment when you compare the two groups of processes. So, having said that, everyone goes through some interesting pains and gains in thinking about, say, White Eddie cheese, White Eddie Creamery, with James and Johnny up there at Karaka. They've had their learning curve was massively steep, and they've just done amazing things. I mean, what people have done in getting going has just been incredible in many instances. And I think also of James Claremont and with Pohiwa Farms in Hawke's Bay, James Thompson and his lovely family in Canterbury. These are real pioneers that have pushed on and really done the hard yards and done some incredible things with what they've got in front of them. So it's a really encouraging. I think what thing comes through from me is this was a very encouraging piece of work, and gave me some real confidence. We've got some fantastic people in the community who really have some brilliant skills, and they brought that to this industry because they can see some future in it. And so it was uplifting, and hopefully it comes through in some of the comments and the things that we've come up with here.
Yeah, thanks, Craig. So in terms of, that's a bit of a snapshot of some of the early findings to sort of try and fit the time. In terms of next steps, we're working on producing a full report of our findings and the survey results. Hopefully that should come out relatively shortly. We definitely see this as a really important potential sunrise industry for New Zealand. So we would like, if we can, to repeat this at least on an annual basis, hopefully, if possible, because we'd like to keep this conversation going. And we think it's important to keep the information to support both new entrants and existing owners in the industry. And lastly, I just want to say again, thanks so much to all those farmers that helped us out with the survey.
Brilliant. Thanks, Geoff. Appreciate that. That was a good summary.
Thank you both, Geoff and Craig. That's really good information and data that we can all, that is really useful for all of us and for those who are not yet in the industry.
All right, so stepping back into my role as the co-chair of this event, we've come to the end of the formal presentations, but we do have a short kind of final comments from Sarah O'Connell, who is there. She is there, and she's going to, and it's back to front, which is, she's going to make a few final comments. So, but yeah, thanks very much to everyone that's come forward and presented today. It's been fantastic, and we had a few little glitches to start with, but we seem to have ironed those out. And can I just say a final thanks to Chris and Callum, Chris Morton and Callum from EventTech, who have been working feverishly in the background to really make this work, as we've broadcast from Zoom out through their systems and out into YouTube land that you've all been listening to. As I said, we're going to be producing this as a set of short videos, and they will be up over the next little while. But as a way of some final comments, I'll introduce Sarah O'Connell. Sarah runs a, well, she can introduce herself, but thanks very much, Sarah, for saying a few words as we go out this afternoon.
Cool, no problem. Thanks very much, Craig. And also thank you to Linda and the rest of your team for pulling the conference together in the way that you were able to, with the changes that had to be made. I think to get everybody on board, from the presenters and the attendees to, tap in virtually to the conference has just been absolutely fantastic. So well done to you, and well done to the presenters for pulling it all together. So as Craig said, I'm Sarah. I work for the AgriBusiness group, and you're probably wondering how on earth I got to be here to give the closing remarks of the conference. I was first introduced to sheep milking when I met Heather McMillan at a Understanding Your Farm Business course. It was probably about 3 years ago now, and that piqued my interest to start with. And then I found out that people were milking sheep in Canterbury, and I touched base with Guy and Sue Trafford, who many of you will know well, and found out more about what their passion was and what they were going to do in their retirement. So watching how they've progressed over the last few years has been really, really great, and watching everything grow. That then developed into me facilitating a Red Meat Profit Partnership action group just with sheep milkers based here in Canterbury, and we've been going for about 12 months, and it's going really, really well. And then that grew into me taking on the position of chairperson of the newly formed Canterbury Dairy Sheep Association, where we've come together to help provide some guidance for new people coming into the industry, and just be a focal point for anybody that wants to know a bit more, and be one connection place for everyone. So that's a little bit about how on earth I ended up sitting in this position, to say farewell to everybody and thank you. I think that the breadth of information that we were shown and talked about today was just superb, and thank you everyone for bringing that information to us all. A number of the key points that I took out from that was from early on with the markets and the international work that Food South have done Our export opportunities are likely to be in that premium space. So let's work hard to ensure that we've got premium products going into that premium space. Nitrates. Mike Joy talked to us a lot about nitrates and how that's a really big issue, especially here in Canterbury, and how it must be considered within our operations and what we're going to do. And, you know, we should be farming to our environment and use the environment to our advantage, no matter where we are in the country. So pointing out, knowing what's good and how we can get the best out of it, I think is really important. We then had a look at sort of the processing and hygiene and farm and the ability to reduce that bacterial growth within our products that can make a huge difference to the quality of the products that are going out. And if we want to get into those premium markets, then we have to have premium products. So ensuring that that supply chain is just flawless all the way through is a really, really important factor. And the technology that's now going to be sitting behind all of that, from the drying, the new drying opportunities and the new freezing work that's been going on, the research in those spaces will provide some opportunity for expansion for smaller scale operations to actually get involved and then scale up to larger as they go on. The session on nutrition, that was kind of mind-boggling for myself. I'm not a scientist, so to be honest, some of it went over my head, but it's really good to know that we've got some top scientists working in this space to really understand the nutritional compounds of our sheep milk, what is best, what's good about it, what might not be so good about it, how does it affect our diet, what's good for it, and how all of that is going to work going forward. So awesome amount of work going on in that space, keep that up. Then we looked at some breeding values, you know, what is the difference of genetics going to make to the national flock and therefore quality of production and quantity of production, and just keeping the whole industry going and moving it forward. And, you know, pick the top of your crop was probably the main point that I took out of that. Do your recording and pick the best. And then we sort of wrapped it up at the end there with a bit of an insight into what our current producers have been doing. How have they been finding it? What's their production been like? What do our country's stats look like so far for our sheep milk industry? And to see that it is growing is really, really good. And that we know that it will only grow further from here. So it's really awesome to see that there's a lot of passionate people involved in the industry. So I think working together is a key point. Everyone out there in the presentations today all kind of had some kind of link to one or two of the other presentations, which is really good. So everyone's got opportunities to collaborate and work together, and gaining traction in markets and is everything that we all need to be working on together. And from a Canterbury Dairy Sheep Association, we sort of, you know, we would welcome any discussions from our North Island counterparts about being able to expand production down here and take the opportunity while the growth is still going here in Canterbury, and while there are some keen and interested players. So on that, thank you all very much for attending. I hope you got some value out of it. With the presentations being available later on, you will be able to go back and read them and listen to them again, and you'll learn something new and you'll reinforce other things that you heard today. So thank you, Craig. Thank you, Linda. Thank you to all the presenters for doing your bit, working with the technology to make it all work for today. I've really enjoyed it. I have sat at my computer all day and watched every presentation, and it has been fantastic. So until the next conference, whenever that is and wherever that is, stay safe, stay well, stay home. Thank you. Goodbye.
Thank you so much, Sarah, for this excellent summary of today. And I just also want to just say a big thank you, not just to my partner in crime, Craig, here, but also to our tech guys who are working behind the scenes, but most of all to everyone who has come in today. There's been a steady number of about somewhere between 140, 160 people. So that just shows, even in times of crisis, this industry is strong, and we're connected, and we're we're staying together and supporting each other. So yeah, and at least the best thing today has been to, for a few hours, not having to think about COVID-19.
Precisely. Thanks for that, Indy. It's Linda. So just a couple of points going forward. I just want to mention that both Maui and Spring Sheep in the North Island will be having open days later in the year. Hopefully we will have recovered from our enforced detention by then, and everything will be ready to roll out. We have, plan to run the conference next year at Lincoln. Do it in such a way that hopefully we'll be face to face and enjoy all the other benefits that can come from getting together as a community. So I've plugged that in for next year around about the same time, but we'll be in touch as we go forward. And just finally, presentations will be up sort of over the next day or so. We'll get them up and people can have a look at the recordings and the video as we go forward. So thanks everyone, and really fantastic to have you all along, and we pulled it off, yay. Despite a few little hiccups along the way. But again, thanks to Chris and the crew, and all the best. Ka kite. We'll see you in the future. All the best. Thank you.
Bye.